Hello everyone, this is another presentation by me. This week is on fluorouracil or 5-FU. Just a little background on what this medication is. It was made in 1957 and 5-FU is a widely used anti-cancer drug that plays an important role in colon and breast cancer, among other cancers such as cancers of the head and neck. There's also a topical version of this that is used to treat skin cancers, which is what we use in the clinic that I work at. It's a topical chemotherapy, basically. And it's just used to kind of help destroy those cells before they grow too far into skin cancers that have to be excised or require radiation treatment. 5-FU is a heterocyclic aromatic organic compound with a structure similar to DNA and RNA's pyrimidine molecules like uracil and thymine, which is why it's an analog of those, with an atom at the C5 position instead of hydrogen. The structure of 5-FU allows it to interfere with the nucleoside metabolism. It can be incorporated into RNA and DNA, and this can lead to cytotoxicity and cell death. Clinical applications of 5-FU have been limited due to drug resistance, with only 10 to 15% of advanced colorectal cancers providing a response to the treatment once it's administered. But in combination with other anti-tumor drugs, this has actually increased the response to 40 to 50%. So by itself, it's not, it doesn't enact too much of a response, but if it's combined with other drugs, it does make a better response from the cancers that it's being treated for. Understanding the mechanisms of how tumors become resistant to 5-FU will greatly help with the understanding of overcoming this resistance and of the development of new microarray techniques. Microarray techniques has offered a chance to determine which genes have roles in producing this drug resistance to 5-FU. Just some side effects of fluorouracil: mouth inflammation and sores, sore throat, esophageal inflammation, diarrhea, loss of appetite, nausea, vomiting, low white blood cell counts, hair loss, skin rash. It can also cause severe toxicity at different levels, and this has to be observed and monitored in order to avoid this, because they have levels from 1 to 4. At a level of about 3 to 4, it can be considered lethal, so it just has to be monitored in order to make sure that the dosage is being controlled, because it is an intravenous drug. You just have to monitor this and make sure that the patients are not experiencing severe toxicity, because it can be fatal if left untreated. The mechanism of action for 5-FU is that 5-FU is converted to fluorodeoxyuridine monophosphate in mammalian cells. This allows a stable complex to form with thymidylate synthase, which then inhibits deoxythymidine monophosphate production. The DTMP is essential for DNA replication and repair, and the depletion of this causes cytotoxicity in a cell. DPD-mediated conversion of 5-FU to dihydrofluorouracil is the rate-limiting step of 5-FU catabolism in normal and tumor cells, and about 80% of 5-FU that is administered is broken down in the liver by DPD. TS is an essential enzyme for catalyzing the biosynthesis of thymidylate and is implicated in the regulation of protein synthesis and apoptotic processes, which is cell death. New research has indicated that 5-FU exerts its anti-cancer effects mainly through the inhibition of TS, and the reduction of the DTMP leads to the depletion of downstream deoxythymidine triphosphate, which then induces perturbations in other deoxynucleotide levels. These imbalances are thought to severely disrupt DNA synthesis and repair, which results in lethal DNA damage. And then accumulation of DUMP can be misincorporated into DNA, and the metabolic product of 5-FU has the same action as this. And since 5-FU is a pyrimidine analog, which means that it can, it's almost the same as uracil or thymine, it can be misincorporated into RNA and DNA in the place of uracil or thymine. And this interference with the normal biosynthesis or function of nucleic acids is therefore another possible mechanism or function of nucleic acids, which is also a possible mechanism of action for 5-FU. In mammalian cells, 5-FU toxicity occurs when 5-FU is misincorporated into the DNA of drug-treated cells, and 5-FU accumulates in the genome rather than uracil extraction. And it can also be misincorporated into RNA, and evidence has shown that RNA-based effects have a significant role in this cytotoxicity. When experiments in yeast have been done, it shows that the defective RNA exosome subunits RRP6P can cause hypersensitivity to 5-FU, and other research shows that certain DNA repair mutations can cause other sensitivities to 5-FU-induced DNA damage, while another mutation causes hypersensitivity, and this is due to RNA-based effects of 5-FU. These results suggest that ribosomal RNA maturation is an important target for 5-FU, along with 5-FU showing enhanced exosome-dependent accumulation of polyadenylated ribosomal RNAs. And then other evidence shows that 5-FU inhibits the splicing of prim RNA through its effect on pseudorylation of snRNA. And then 5-FU is also inhibiting the processing of pre-RNA into mature ribosomal RNA, while also disrupting post-transcriptional modification of transfer RNAs and the assembly and activity of snRNA and protein complexes, which then inhibits pre-mRNA splicing. RNA that contains 5-FU can also inhibit pseudorylation, which is the most abundant post-transcriptional modification of non-coding RNA. And this little diagram down here is just showing you how 5-FU is introduced to a cancer cell, and then how it goes to the immune cells and kind of shares this information with them so that they can also fight off the cancer cells so that they do not continue to replicate and divide in the body. This picture is actually a side effect of like the topical 5-FU. It's called Efudex, and this is the topical chemotherapy that I talked about earlier. There can also be this kind of like redness reaction at the injection site where the intravenous FU is introduced in, but this happens over time. It's not super common, but the topical chemotherapy creates a much bigger reaction on the skin. And this is showing the inhibition of the thymidine, and just shows how it disrupts like DNA replication and repair and causes DNA damage. This is the molecular structure of 5-FU, and then this is a very large diagram that just shows one of the mechanisms of action of 5-FU. So you can see we've got our signals with the receptors here, and then just how it kind of acts within the cell. And then these are the sources that I use to get my information. I hope you enjoyed this presentation, and thank you.